Assalamu alaikum dear students, hoping you all will be fine. Today I will continue from where we left our lecture. हम लोग लिपिड्स के बारे में पढ़ रहे थे। मैंने आपसे कहा था कि लिपिड्स की बहुत सारी टाइप्स हैं, जिनमें से मेजरली हम इसको दो हिस्सों में तकसीम करते हैं। एक हमने कहा था कि हम इसको डिवाइड करते हैं इन दी स्टोरेज लिपिड्स, और दूसरा मैंने इनको कहा था कि स्ट्रक्चरल लिपिड्स हैं, फिर सिग्नलिंग लिपिड्स होते हैं। Initially, we will start from the storage lipids. The example is triglycerides. I'm sorry, example is triglycerols. Triglycerols are non-polar compounds. Majority of the fatty acids in the biological systems are found in the form of the triglycerols. They are of two types. One is in the solid form, to some extent the semi-solid form, and the second one is the liquid form. The semi-solid or solid form are known as the fats. The liquid ones are called the oils. These are the primary storage form of the lipids, uh, mostly known as the body fat. These are less soluble in water than fatty acids due to the lack of charged carboxylate group. Less dense than the water, fats and oil can float on the water because of their less density. This is the structure of the triacylglycerol. The major compound is the glycerol compound. Glycerol compound has the three positions where the hydroxyl group is attached. These hydroxyl group can be replaced by the or can be esterified with the you can say these can be esterified with the fatty acids. You must be clear between the term that there is the ester linkage with the fatty acid group. Ester is the link where the one carbon, where the linking carbon is attached to the oxygen, and it is also attached with another oxygen. You can see the linking group. So, for example, this is the carbon and this is the carbon. The linking group is attached with the two carbon atoms, with the two oxygens. Here the oxygen, and second one is the here is the oxygen. There are two oxygen involved in this linkage, are present in this linkage. So the linkage will be the ester linkage. Linking triacylglycerol can be of same type, if these are of same type, the the linking fatty acid, if these are the same groups, then the simple word tri is added. Say for example, on these three positions, only palmitic acid is attached. Then this molecule will be known as the tripalmitin. But this is not the case always. These fatty acids can be different on the different positions. Say for example, um, on position number one, the other group is attached. Position two, other group is attached, and position three, another group is attached. So such kind of triacylglycerols are known as the mixed triacylglycerols, and these are named according to the presence of the fatty acid on the glycerol, where these are present. Say for example, in the example given, say for example, on position number one is stearoyl, on the position number two is linoleoyl, and on the position number three palmitoyl. So it will be named as the 1-stearoyl, 2 linoleoyl, and 3 palmitoyl glycerol. This is a mixed triacylglycerol. Fats can provide the efficient fuel storage. The advantage of the fats of the polysaccharide that fats can carry more energy per carbon because they are more reduced compounds as compared to the carbohydrates. Fatty acids carry less water per gram because they are non-polar. Uh, for example, glucose and the glycogens are for the short-term energy needs. They have the quick delivery. Fats are for long-term. For example, for the months, these are stored to provide the energy, and their delivery is very slow. What is the reason behind this? What are the advantages and disadvantages? The major disadvantage is that the fat cannot provide the energy on the real time, on the spot when the energy is required, because of their issues in the transportation, because fat cannot be dissolved in the water, so they cannot be transported by the water simply. So they need some carrier proteins to transport them. As well as the carbohydrates are concerned, the carbohydrates are soluble in water, and they can be transported with the movement of the water from one position to the other position. So this is the major, you can say, the disadvantage. But the advantage is these compounds are more reduced compound and can provide the high amount of energy. They can provide the high amount of energy. You can say to some extent the double as compared to the carbohydrates. But this is true that uh, if we store the same amount of carbohydrate, and the, you can say, if we have if we have the carbohydrate can provide the more energy as compared to the carbohydrate can provide less energy as compared to the lipids. This is the diagram of this fats provided efficient fuel storage, triacylglycerols. The next example is waxes. Before going to the waxes, some functions of the triacylglycerol. TG provide the stored energy. It can provide the insulation to the cells. Or it is it can provide the energy, and it can provide the insulation. In the eukaryotic cell, TG form separate microscopic phase of the oily droplets in the aqueous cytosol as the deposits of metabolic fuels. In invertebrates, there are some specialized cells which are called as the adipocytes or the fat cells. They are responsible to store the large amount of the TG as fat deposit droplets. While in plants, the fats are stored in the seeds as oil. Energy provided for and these are stored in the seed, and they provide the energy for the seed germination. Adipocytes and germinating seeds contain lipases, the enzyme that catalyzes the hydrolysis of triacylglycerols and release fatty acids to export to the site where they are required as a fuel. Advantages of using TG as a stored fuel. There are as a stored fuel rather than the polysaccharide glycogen and the starch. There are two major advantages. Number one, triacylglycerols are the hydrophobic compounds. These are unhydrated, so organisms have triacylglycerol. These are the hydrophobic, so unhydrated. So organisms having fat as a fuel does not carry extra weight of water for hydration associated with the stored polysaccharide. Um, इसका मतलब ये है कि ट्राइसल्गलाइसरोल जो हैं, these are the हाइड्रोफोबिक कंपाउंड, इस वजह से इनको एज अ फ्यूल वर्क करने के लिए वाटर की ज़रूरत नहीं पड़ती. अगर इनके लिए वाटर की ज़रूरत पड़ती होती तो एक्स्ट्रा वेट ऑफ वाटर कैरी करना पड़ता. Carbon atoms of the fatty acids are more reduced than the sugar or the carbohydrate. Oxidation of the fat, because of this reason, oxidation of the triacylglycerol yield twice times energy as compared to the oxidation of the carbohydrate. Human has um, Human have fat tissues, adipocytes under the skin, abdominal cavity, mammary glands, etc. Moderately obese people with the 15 to 20 kg carrying triacylglycerol in adipocytes um, for months. If an obese person who is carrying the weight of 15 to 20 kg of the triacylglycerols, um, could meet the energy requirement for the month as compared to the carbohydrate if stored in the same weight. यानी कि अगर 15-20 kg किसी बंदे की फैट्स में अगर किसी बंदे की बॉडी में अगर फैट्स डिपॉजिट हों तो वो तकरीबन मंथ तक गुजारा कर सकता है उन फैट्स पे एज कम्पेयर टू दी पर्सन जिसके ब्लड में इसी सेम अमाउंट में अगर कार्बोहाइड्रेट्स आ जाएं बिकॉज कार्बोहाइड्रेट इसी अमाउंट में लेस देन अ डे काम कर सकते हैं इससे ज़्यादा नहीं. In animals, because triglycerides are stored under the skin, they serve as the energy store and they provide insulation against the low temperature. And these are also present in the seals, walruses, penguin, and warm-blooded animals, and provide them the insulation against the low temperature. These are present. These are also present in the hibernating animals, and in the hibernating animals, the fat serves as accumulated insulation storage and serve for the maintenance of their life. You can say in the hibernating animal, they provide both of the function, insulation and the storage for their survival. Sperm whales, tricyclisaur, and waxes are present. They help in buoyancy during the deep dives. I'm saying that in the deep diving animals, they provide them the buoyancy. Buoyancy means okay, they can float in the water. Sometimes to increase the shelf life of the cooking oil, the partial dehydrogenation process for the triacylglycerols or the natural fats is done. Uh, because of this partial dehydrogenation, because of this partial hydrogenation of the cooking oil, the trans fats are produced. The natural fats, say for example, vegetable oil, dietary fats, and the animal fats, and the complex mixture of these natural fats are the complex mixture of simple or the mixed triacylglycerols. They contain variety of fatty acids and they contain variety of chain length and they have different degree of saturation. Vegetable oils in the vegetable oils mostly TGs are unsaturated. TGs contain the unsaturated fatty acids, so that's why these are in the liquid form. Saturated fatty acids like tristyrene, white, greasy, solid at the room temperature, and this is present in the beef fat. Lipid-rich food exposed to the oxygen in air spoil these fats and produce unpleasant smell and unpleasant taste. Um, so this is because of the result from the oxidation. You can say this is because of the result of oxidative cleavage of the double bond. To reduce this to reduce the spoilage of the fats, mostly the partial dehydrogenation of the fats is done. Because of this partial hydrogenation, sometimes fat, instead of becoming hydrogenated, only their double bond conformation is changed. It means sometimes it changes from cis form to the trans forms. When the fats are transformed into their trans forms, then such kind of fats are not good for the health. And they cannot be digested easily, and they are involved in producing several different types of diseases. Waxes are the esters of the long chain saturated and unsaturated fatty acids with long chain alcohols. These are insoluble and have high melting points. They have a variety of functions. In the planktons, they can be used as the planktons can use them as a metabolic fuels. They can provide protection and pliability for the hair and skins in the vertebrates. For the bird, they provide waterproofing of the feathers. In the tropical plants in the ivy, they provide protection against evaporation, and they are also used in the lotions, ointment, and polishes you can see in the cosmetic products. Beeswax is a mixture of a large number of lipids, including esters of triacontanol and long-chain alkane, hentiacontan. Our next topic will be the structural lipids. Waxes are very important for the diving birds because their feeders contain the waxes. Because of these waxes, they can their feeder because the waxes are the hydrophobic compounds, so the water is not stick to their feeders. So water cannot interact with the feeders. So they can dive and come back from the surface of the water, can back from after diving from the water. If the waxes are removed from their feeders, they can dive in the water, but they cannot come out of the water. So waxes are very important because their feeders will be wet and they cannot be at the weight of the water and cannot fly. So waxes are very important compound. Waxes are very important for their feeders. This is the end of today's story. Hoping, inshallah, I will join you tomorrow with these structural lipids. Good luck.